Hello friends, this is Dr. Sachidanand Singh, Faculty of Biotechnology. So, in our previous lectures, we have understood about protein structure prediction and its analysis, and we have went through n number of tools, databases, algorithm and the steps and processes. We also tried to understand what are the different approaches by which protein modeling and structure prediction can be done. We also saw few validation softwares and we did online performance to see how for a specific protein analysis can be done. So, this all was going in different exercises and our previous lecture. Now, onwards we are seeing specific online server or databases to understand how protein structure prediction or analysis can be done. In this approach, let us today understand another web server known as PROSA. So, let us go ahead with PROSA and see that how protein structure prediction can be done, what are its different analysis, which algorithm it contains, and all this process we can go ahead in this lecture series. So, let us start with this. PROSA web, it is a protein structure analysis component. So, let us see the introduction. PROSA web can be broadly explained as protein structure analysis. It is a web based tool widely used in the field of structural biology and bioinformatics. The course which we are studying is a structural bioinformatics course pertaining to protein structure prediction and analysis. It serves as an essential resource for evaluating and analyzing the quality and reliability of protein structures determined through experimental or computational method. So, what we are going ahead here is the computational method. PROSA web aids researchers in assessing the plausibility of protein structures by comparing them to a vast databases of experimentally determined protein structure. So, when we say vast databases of experimentally determined structure is PDB, yes, in protein data bank the structures which has been submitted just like NMR, nuclear magnetic resonance data structures, and by X-ray crystallography. It helps to identify potential errors or discrepancies. So, let us see a brief overview of PROSA web server. It offers a straightforward and user friendly interface that allows users to upload a protein structure file in various formats, and in our previous lectures we have already seen the PDB structure which has been submitted is in default in which format? Yes, it is in PDB format. And what is PDB? Protein data bank, and where it is been located? It is been given by Rutgers University. So, PROSA it performs a comprehensive analysis of the input structure, assessing its compatibility with known protein structures and generating insightful graphical presentations for visualization. So, that is about a brief overview of PROSA. So, let us see from where it got originated. PROSA is a web based implementation of protein structure analysis software. It was developed by Wolfgang Kabsch and Chris Sander. The PROSA method was initially introduced in early 1990s as a tool for assessing the quality of protein structure determined by X-ray crystallography. Over the years, it has been refined and adapted to include protein structures determined by other experimental techniques as well, which we have already seen, such as NMR spectroscopy and computational modeling. The transition to a web based platform expanded its accessibility to researchers worldwide, so that we can actually go ahead and understand whatever proteins we are developing by our computational method. So, what is it? Firstly, it was explaining the X-ray structure, then NMR structure, and then what we started working on, computational based methods. So, now let us see the application of PROSA web. PROSA web server is has a variety of applications in structural biology and its related field. So, let us see the first application. It helps in quality assessment. It provides a rigorous assessment of the quality and reliability of protein structures, helping researchers to identify potential issues and errors. The second is model validation. PROSA web server can be used to validate and refine protein structure models generated through computational methods like homology modeling or ab initio modeling. Structure comparison. Researchers can compare the quality of multiple protein structures to choose the most reliable one for further analysis. Third one is database integration. PROSA web is integrated with the protein data bank, that is PDB, allowing users to access a comprehensive database of protein structures for comparative analysis. PROSA is also used in educational category. It serves as an educational tool helping the students and researchers learn about protein structure assessment and quality criteria. Many courses includes this as an experimental category, and people teaches through PROSA web server about protein structure prediction. So, let us see now what are the steps to use PROSA as in web server. So, it includes following step. The first thing is upload the protein structure which has been developed either by computational method, either by NMR understanding or X-ray crystallography. Users have to upload their protein structure file in PDB format through the web interface. Second, what PROSA will do is PROSA will start initiating structural analysis. PROSA web performs a comprehensive analysis of the input structure using established algorithm. So, there are different algorithms behind this. Let us see that. The first is generation of z-score plot as a statistical analysis. The tool generates a z-score plot which is a graphical representation of the input structure to calculate its energy score compared to a database of experimentally determined structures. Z-scores measures the structural deviation for the expected value for a typical naive protein structure. So, that is a statistical analysis as a plot diagram in z-score calculation it will show. Second is visualization. Users can visualize the z-score plot and identify potential outliers, the regions which are not obeying the Ramachandran plot, potential outliers or regions of concern in the protein structure on which further little more refinement can be performed. Further, it gives an analysis report. PROSA web server generates a detailed analysis report that includes information about the z-score, potential outliers and regions of the protein structure that may require further investigation. What do you mean by further investigation? To go for more refinement. In our previous lecture, we have already shown that further refinement can be done through loop modeling and other approaches. So, let us see how it works out. PROSA web employs a sophisticated methodology of protein structure analysis. So, the first thing what it does is energy calculation. The tool calculates the potential energy of the input protein structure using a knowledge based potential energy function. What is that? Knowledge based energy function based on potential energy. This function relies on statistical information derived from a large database of experimentally determined protein structure. And whenever we say experimentally determined protein structure is, we have to remember yes, PDB, and what we have to say in PDB through X-ray crystallography and NMR. Then z-score calculation is done. So, let us see how it is done. The calculated energy of potential energy is then transformed into a z-score which quantifies how far the input structure deviates from the expected energy distribution of naive protein structure. So, it is a kind of seeing the deviation or root mean square deviation. Then it performs statistical analysis. PROSA web server statistically evaluates the z-score calculation to assess the quality of the protein. So, a higher z-score indicates that the structure is more deviant from the typical naive structure, potentially suggesting errors or inaccuracy. So, this means the lower the z-score value, the better is the protein structure, right. So, if we see, we have the most important aspect of PROSA web server is to understand the z-score calculation. By the graphical representation, we can justify our results in a better aspect. So, let us now see the advantages of PROSA web server. The first one is, it is a graphical user interface. So, it is a user friendly. It is a web based interface which is accessible to a wide range of users without requiring specialized software installation. So, it is a web based online. Second, it gives a very comprehensive analysis. It provides a thorough evaluation of protein structures, highlighting potential issues and errors. The third one is, it gives a quality control. Researchers can use PROSA web to ensure that the protein structures they work with are of high quality and reliability. Third, it gives visualization, a better visualization aspect. The z-score plot and analysis report offers a intuitive visualization for quick interpretation of structural quality. And finally, it gives us the educational value. As we have already seen, it serves as an educational tool for teaching and learning about protein structure quality assessment. So, there are n number of challenges also, because when it is a server and it has passed through the CASP competition, but still different challenges, and which the scientists come out of those challenges, develop new algorithm and come out with new aspects. So, what are those challenges? Let us see. While PROSA web is a valuable tool, it has some limitations. Dependence on the input quality. The accuracy of PROSA web analysis depends on the quality of the input protein structure, which type of protein structure we have given and what is the quality, the whole analysis will perform on that. Assessment focus, it primarily assesses global structure quality and may not detect specific local errors. So, it works out as a global protein structure, not at very specific location errors or inaccuracies. Lack of repair features, PROSA web does not provide tool for automatically correcting the errors, like how we saw in modeler, we have to programmatically we have to do loop modeling. Here we can't do the errors correction at their level itself. So, overall we can say, to talk about PROSA in a small brief conclusion, PROSA web plays a pivotal role in structural biology and bioinformatics by offering a user friendly platform for protein structure quality and assessment. Its robust methodology and user friendly interface make it a valuable resource for researchers, educationalists and students. By identification of potential issues in protein structures, PROSA web contributes to the improvement of structural biology research and the reliability of protein structure based studies. As the field of structural biology continues to evolve, PROSA web is likely to remain an essential tool for assessing the quality and trustworthiness of the protein structure. So, this was about a brief introduction about how PROSA and what are the brief steps how it works out. Now, let us take it elaboratively, what are the PROSA steps and why we need a PROSA to have a concise overview and to understand why for protein structure prediction it is important. So, let us go into a detailed explanation of PROSA and how the input files will be, what is the algorithm and what are the level of structures which we can study. So, let us start from this initiative level, that why we need PROSA. So, in PROSA protein structure prediction is done, hence protein is what? Protein are the molecular workhorses of life, performing a wide range of essential functions in living organisms. Understanding their three-dimensional structure is a crucial for elucidating their biological functions and designing drugs to target specific protein. That is what we study protein for. One of the most significant advancement in the field of protein structure analysis is protein structure prediction. In protein structure prediction, n number of tools are been reported. Among them is one PROSA. It is a powerful computational tool that plays a pivotal role in determining the quality and reliability of the protein structure. So, here we will explore the significance of PROSA in protein structure analysis in a detailed view. its understanding principles and its application in various scientific domains as well. So, the importance of protein structure analysis includes, as proteins are macromolecules consisting of long chain of amino acids folded into complex 3-dimensional structure. So, these structures are fundamental to understanding protein functions and dynamics. Knowing the precise arrangement of atoms within a protein can provide insights into its enzymatic activity, binding sites of ligands and potential biological roles. Hence, protein structure analysis is an essential for various scientific and practical applications, which includes drug design, protein engineering and understanding disease mechanism. So, in this context of drug discovery, for example, knowing the 3D structure of a target protein will allow researchers to design small molecules that can bind to specific region of a protein, modulate its function and to modulate and to go ahead for potentially treating diseases. So, these are some aspects when we do a protein structure prediction in drug discovery. The challenges happening in protein structure analysis may be determining protein structure experimentally can be a laborious and time consuming process, often requiring techniques like X-ray crystallography or nuclear magnetic resonance, that is spectroscopy method. These methods have their limitations and not all proteins can be easily crystallized or studied using NMR. Additionally, experimental methods may provide incomplete or low resolution structural information. To overcome these challenges, computational methods have become indispensable tool in the field of structural biology. Computational techniques can predict protein structures with remarkable accuracy and can be used to assess the quality of experimental structures. PROSA is one such computational tool that has made a significant impact on the field. So, let us see then. PROSA is a short of protein structure analysis, is a web based platform and a standalone software tool used for the evaluation and validation of protein structure. As we already saw, it is developed by Wolfgang Kapp and Chris Sander and has since become an essential resource for structural biologists. So, the primary objective of PROSA is to assess the quality and reliability of the protein structure. So, when we talk about quality, it talks about the validation and assessment, and we already saw it accomplishes by assessing or calculating by Z-score, which measures how unusual or deviant a given protein structure is formed with the set of reference structure. A low Z-score indicates the structure is within the expected range of native protein conformation, while a high Z-score suggests potential errors or inaccuracies in the structure. So, what is the principle? PROSA calculation of Z-score is based on statistical potential energy function. This function estimates the energy of a given protein structure based on the pairwise interaction between the constituent atoms. The energy is compared to the average energy of a set of experimentally determined protein structures with the similar size, so that the energy calculation can be done. The Z-score is then calculated as the deviation of the target protein energy from the mean energy of the reference structure, normalized by standard deviation. The key principles behind PROSA can be summarized as follows. Statistical potential. PROSA uses a statistical potential derived from a large data set of high quality protein structures to estimate the energy of a given structure. Second, deviation assessment. The Z-score quantifies how far the energy of the target structure deviates from the mean energy of the reference structure. So, the mean calculation is being performed. A low Z-score indicates well folded native like structure, while a high Z-score suggests potentially structural issues which need to be again reframed. In graphical representation, PROSA provides graphical representation of the Z-score, allowing users to visualize potential errors or inaccuracies in their protein structure. Regions with high Z score are typically highlighted, making it easier to identify problematic errors. So, PROSA finds its application in different scientific domains, specifically in bioinformatics, structural biology, drug discovery and protein engineering. So, it helps in wide assessment of the quality of experimentally determined protein structure, and it helps the researcher to identify potential errors, the uncertainty in the structure, a guiding for future refinement. It also helps in the evaluation. PROSA is employed to evaluate the ranking of the protein structure which are being generated by model generation method and computational modeling technique, which includes the base as homology modeling or de novo protein structure prediction method. So, two algorithms are being considered in PROSA, that is homology modeling and novo protein structure prediction method or ab initio method. Protein engineering is another application by assessing the impact of mutations or modification on protein stability and structure. This information is very critical for designing protein with desired properties. It also helps in drug discovery, specifically with structure based drug design. Drug discovery is divided into two portions, that is ligand based drug design and structure based drug design. So, PROSA helps in structure based drug design where the target protein is important to understand its structure and active site. So, structure based drug design relies on accurate protein structure. So, PROSA assists in validating target protein structure for virtual screening and the rational designing of small molecule. It also helps in disease mechanism by understanding the structural basis of disease related protein, and which is very crucial for drug development and disease mechanism. PROSA helps researchers to evaluate disease associated proteins as well. So, let us see now input tools of PROSA. While PROSA is a valuable tool, it has some limitations as well. It depends upon the reference data. PROSA relies on a data set of experimentally determined protein structures which may not cover all the possible protein conformation. Hence, the quality of result can be affected by reference data set. It is a very sensitive to model size. The Z score calculation can be a sensitive to the size of protein structure being analyzed. Smaller the protein may yield higher Z score even with the accurate structure. It has a limitation to identify local areas or loops where any problem has happened, and it is not able to detect the errors for the specific thing. It also is not able to do the local error calculation as well. So, now let us see the future development in PROSA. So, future development in PROSA and related tools will aim to address these limitations. Researchers are working on improving the accuracy of statistical potentials, expanding reference data set. The reference data set can be enlarged so that it can go ahead and get the related information. So, PROSA web is basically, to understand in overall, is an interactive web service for the recognition of errors in 3-dimensional structure of protein, and the availability of structural model of a protein is one of the key for understanding the biological process at a molecular level. The recent advances in experimental technology have led to the emergence of large scale determination pipeline aimed at the rapid characterization of protein structure. The resulting amount of experimental structure information is enormous. The application of computational methods for the prediction of unknown structure adds another plethora of structural model. So, same like that, the latest NAR web server, NAR web server example, 50 tools are there in this category of 3D structure prediction is also one kind of part. The assessment of the accuracy and reliability of the experimental and theoretical models of protein structure is a necessary task that needs to be addressed regularly, and in particular it is essential for maintaining integrity, consistency and reliability of public structure repositories. PROSA is a widely used check tool for 3D models of protein structure for potential errors. Its range of application includes error recognition in experimentally determined structures, theoretical models in protein engineering. PROSA web encompasses the basic functionality of standalone PROSA and extends it with new features that facilitate interpretation of the results obtained. The overall quality score can be calculated by PROSA for a specific input structure which is displayed in a plot, and that shows the score of experimentally determined protein chains which are currently available in PDB. This feature relates the score of a specific model to score computed from all experimental structures deposited in PDB. Problematic parts of a model are identified by a plot of local quality scores, and same score are mapped on a display of 3D structure using color code. So, by the color code we can understand it clearly. A particular intention of PROSA web application is to encourage structure depositors to validate their structure before they are submitted to PDB, and to use the tool in early stages of structure determination and refinement. The service requires only C alpha atoms so that low resolution structures and approximate models obtained early in the structure determination process can be evaluated and compared against resolution structures. The PROSA web server returns results instantly. At once when you submit, the algorithm will run. So, let us see the required input as a web server usage. PROSA requires the atomic coordinates of the model to be evaluated. So, how a user can use it? User can supply coordinates either by uploading a file in a PDB format or by entering the 4-letter code of the protein structure. A chain identifier and NMR model number may be also used as a specify a particular model. A list with possible values of this parameter is presented to the user if the entered chain identifier or a model number is invalid. If no chain identifier or model number is supplied by the user, the first chain of the first model found in the PDB file is used for the analysis. So, now let us see, after uploading the file into the server, what are the range of computations. The computational engine used for the calculation of score and plot is standard PROSA, which uses knowledge based potential of mean force to evaluate model accuracy. All calculations are carried out, what we saw before is C alpha potentials. Hence, PROServa can apply for low resolution structures or other cases where C alpha trace is available. Only a set of C beta potentials is included in the stand-alone version of PROSA. After parsing the coordinates, the energy of the structure is evaluated using a distance based pair potential and a potential that captures the solvent exposure of protein residues. From these energies, two characterization of the input structure are derived and displayed on the web page. The first is Z score and the plot of the residue energies, which we have already seen in the previous up slides, that how the Z score is been calculated. So, when the Z score is obtained, it indicates the overall model quality and its understanding. The Z score outside a range characteristics of the native protein indicates an erroneous structure. In order to facilitate interpretation of the Z score of the specified protein, its particular value is displayed in the plot. In the plot we can understand. So, that contains the Z score of all the experimentally determined protein chains in current PDB. Group of structures from different sources, X-ray, NMR, are distinguished by different colors. The plot can be used to check whether the Z score of a protein is under the applicable component or not. The next one, we will receive a energy plot that shows the local model quality by plotting energies as a function of amino acid sequence position as a notation as I. In general, positive values corresponds to a problematic or erroneous part of model, wherein a negative value is a stable one. A plot of single residue energies usually contains large fluctuations and is of limited value for model evaluation. Hence, the plot is smoothened by calculating the average energy for 50 residue fragment. So, ith plus 39, 40 amino acid sequence will be calculated. in order to further narrow down those regions in the model that contribute to a bad overall score, PROSA web visualizes the 3D structure of the protein using molecular viewer tool, and the very genuine molecular viewer tool is JMOL, which can be identified from the URL mentioned here, http://www.jmol.org. So, JMOL is an open source tool where visualization can be done in a very best way and color coding can be represented. Residues with unusual high energy stand out by color from the rest of the structure. The interactive facilities provided by JMOL like distance measurement and energy calculation can also be reported. Protein structure validation can also be performed into this portion. So, a PROSA is a tool which provides typical example of application of PROSA web in the validation of protein structure. It also helps in giving the Z-score value, it also helps in giving the visualization tools. So, in the PROSA web server there are help file where different examples are given, or we can perform by giving our own PDB ID and the whole analysis can be performed. So, by this overall analysis, we saw a very detailed understanding of PROSA, its steps, its inbuilt process, its algorithm and its overall results and analysis in graphical representation. So, that all gave us a very good overview, and this PROSA web based tool can be used by scientists and teachers to take it as one of the experimental lab and to explain the students the better representation and understanding of statistical analysis. So, overall let us wind up with understanding PROSA in a concise way. It is a diagnostic tool that is based on statistical analysis of the available protein structures. The potential of mean force compiled from the database provides a statistical average over the known structure. Structure of soluble globular proteins whose Z-score deviates strongly from the database average are unusual, and frequently such structures turn out to be erroneous. For protein containing membranes spanning regions, the significance of deviation from the average over the database is less clear. Hence, these are some approaches where PROSA can be used, where for students can utilize by putting a 4-letter code ID and can analyze this whole PROSA server step by step. So, this was all about PROSA. Now, we understand. So, now this is all about PROSA to understand a protein structure by X-ray crystallography, NMR or computationally modeled structure, and to see the statistical significance by understanding the Z-score value and energy calculation. So, this tool gives an overall view of protein structure prediction and understanding statistical parameters. So, by putting up a computational model protein, a significant graphical representations come out of this tool, which can be reported into thesis or significant articles for publishing. So, in the coming lecture, now we will understand how protein model can be done or protein validation can be done through what if server. Thank you.